Ready. All right. Well, hi everyone. So my name's Kassan Pine. For those of you that I don't know, there might actually be a few people here I don't think I've met. So I'm Kassan. I finished my master's here at Guelph in December in the Department of Integrative Biology, where my thesis focused on kind of bioinformatics, evolution, and genomics. So I have a ton of genomics experience, not just with my master's, but also some other work I'm doing on campus right now, more in like the eDNA metabarcoding space. But yeah, so I have a lot of genomics experience, and I think that lends well to what I'll talk to you all about today. And so I joined the GBADS informatics team about 2 to 3 months ago, I believe. So my project has been working on metadata mining in NCBI, which is what I'll discuss today. So my plan for the next 30 minutes or so, depending on time, is to kind of start off with a brief introduction on genomics and NCBI to kind of set up the stage for the rest of the talk. And then I'll focus on the project and describe kind of some results and limitations, and then finish up with what I think could be some interesting avenues of future work. So I think it's important to kind of briefly go over some genomics terms before talking about NCBI, because it helps set the stage for some of my results and kind of to get everyone on the same kind of playing field. So as I said before, I have tons of experience with genomics. My master's was more population genetics and genomics. So I was looking at DNA sequences for individual fish, where now I'm working more with like eDNA metabarcoding, which I'll kind of get into some of the differences between all of those in a few slides, but that's kind of my background. So genomics is the study of the function, evolution, and structure of the genome, while genetics is just the study of genes. So as you would expect, genomics and genetics research tends to overlap, which is why large databases such as NCBI house like both types of data. So genomics and genetics research used to be a lot more expensive, as the cost of sequencing, even for a small base pair length, was really high. However, sequencing is now more affordable, not just for model organisms with smaller genomes, but even non-model organisms with more complex genomes. And to kind of give you an example, the fish that I was working on in my master's, their genome, like there was a whole genome duplication event in a common ancestor. So their genome is like double like the size of some other genomes. So in the past, sequencing a genome like that would have been so expensive, and now it's a lot more affordable. I mean, it's still not super cheap, but now that it's affordable, things like this are getting sequenced more often. So this boom in sequencing data is mostly due to high throughput sequencing, like Illumina sequencing, where genomes and genomic data are being sequenced faster than ever before. And I think this is an important point because now that sequencing DNA and RNA is cheaper, there's a ton of sequencing data being produced across so many fields. So like medical genomics, health genomics, and like all the way to like population genomics, where, we were sequencing a weird genus of hybridizing fish that don't have a huge economic, they're not great for, people aren't sequencing those genomes for any economic reason, but more so for like large evolutionary questions. So as you would probably expect, genomic sequencing data are not small files. So this increase in sequencing data has caused a bit of a data storage issue. There's estimates that predict that there is 2 to 40 billion gigabytes of genomic data generated every year. And over the next decade, it's predicted that genomics research will generate between 2 and 40 exabytes of data. So to kind of really drive that point home, I have this infographic here that illustrates this prediction. So you can see how much data will be produced by genomics research, where like each shark is 100 million gigabytes of data. So if you look at this whole picture and imagine this is all genomic data, like it's a lot of data. So with different research groups and organizations across the world generating these data, it's important that there's, some sort of database or repository for these data to be stored, which brings me to NCBI. So NCBI is the National Center for Biotechnology Information. It stores data related to biotech, biomedicine, bioinformatics. So it holds genomic and genetic data, including genomes, as I've mentioned before, gene sequences, expression patterns, protein sequences, and so on. So one of the most used tools that you may have heard of is BLAST. It's a local alignment tool. It's used a lot in like undergrad genetics courses, but basically you can get sequence similarity information for your DNA sequences against the database. So usually when people think of NCBI, they think of BLAST, but NCBI is a lot more than BLAST. And NCBI is funded by the US government, but organizations across the world contribute to it, which is nice because you kind of get a whole bunch of data, not just from like the US and Canada. It was founded in 1988, so there is a large amount of data stored on NCBI, but as sequencing gets less expensive and more affordable, the amount of data is just growing every year. So here's just a quick look at the main page of NCBI. So you can see on the left are just different resources, kind of from chemistry to genetics to taxonomy. In the middle of the homepage is kind of where you would submit, download, or analyze data. I haven't submitted any data myself. I will in like the next month, but I've heard that it's a relatively not super painful experience, which is good. And then on the right are just more tools and resources like BLAST, which is what I was just mentioning before. So in terms of databases, so there's a bunch of them. So these are all of the databases on NCBI. And you can see that they're split up by type. So you have literature, health, genomics, genetics, proteins, and chemicals. And what's nice about NCBI is that many of the databases are linked together within NCBI. And then when it comes to descriptive data, like metadata, there are only a few databases that store this information. And since these data are genomic in origin, most of the metadata is focused on like specific methods. For example, like sequencing platform, whether it's a HiSeq, a NovaSeq, a MiSeq, for example, or like base pair length of sequences and things like that. So there's really only one database that includes metadata for the organisms used in these studies, which is the biosample database. I've also included bioproject in this box, just because the bioproject database links to the biosample database. Like to give an example, if you have an entry for a goat and it's a sample from like a project of 10 different samples, the biosample database would link to that bioproject that would have all of the samples listed. So for GBADS, biosample is really the only database of importance out of all of the databases that NCBI has, because it's really the only one with like descriptive metadata about the organisms used. So to kind of give you an idea of some of the types of data, here are two entries from the biosample database. So I've included two here to kind of show the difference between two main types of entries in the biosample database that I'll kind of touch on throughout the talk. So on the left, there's an entry for a pig DNA sequence, and there's different categories of information included here. You can see identifiers, organisms, attributes, links, bioprojects, etc. And the attributes, I just want to point out, is something that I'll be talking about a lot throughout the talk. So when I talk about attributes, I just mean like the specific bolded kind of attribute names there, like source name, breed, treatment, etc. So on the right, the setup is pretty much the same, except that it says bovine metagenome for the organism rather than just bovine. So this brings up the difference between a genome and a metagenome. So a genome consists of sequences of just one organism, while a metagenome is a collection of sequences recovered from the environment. So metagenomes are common in metabarcoding studies from like eDNA samples, where a sample is taken from the environment, that could be like soil or water, and then it's sequenced for all individual DNA included in the sample. So what this means is that a metagenome sample could have DNA sequences from multiple organisms. So metagenomes in this case is not really representative of the data that we're trying to capture for GBADS, because an entry could have information for multiple organisms and individuals. So my project was NCBI metadata mining. So I queried the biosample database to extract information about GBAD species. So in this case, it was cattle, sheep, goat, chicken, livestock, like things like that. But this project can be expanded to many other species. So I queried attributes like species, weight, sex, age, location, and coordinates, and then date, and health information. And again, just a reminder that attributes in this case were kind of those bolded terms that I showed on the last slide. So the idea behind this project is that metadata from the biosample database on NCBI could be a new data source for GBADS, as we would be able to mine weight, age, sex, et cetera, and even like species list per country, for example, or something along those lines. So before I get into more of the specifics of what I did, I wanted to set the stage a bit about the biosample database, now that you kind of all know that my project focused on mining the metadata. So this figure is actually from a paper that looked at the quality of metadata from the biosample database. And so when researchers upload genomic sequences to NCBI, they can upload their metadata as different packages. And basically what packages mean in this case is like how they can upload the data. So for example, if you use the human package, it would include common attributes that relate to humans. And you could see from this figure that there are packages for most organisms like invertebrates, plants, or even, viruses. And each package in this figure includes the number of entries that use that package and the percentage. You can also see from this figure that most of the metadata entries use the generic package. And what's really fun and interesting about the generic package is that it doesn't require any specific attributes and allows custom attributes. So you can imagine how variable the data are if users are allowed to kind of add their own attribute names, meaning that searching for age, for example, could just be like age or age in years or just years or something like that, or a misspelled version of any of those. So this will kind of follow through in my talk, but I think it's kind of an interesting point to make kind of off the hop here. So now that we kind of get an understanding of how the metadata can be quite variable, here's another figure from that same paper. It's the Goncalves paper that looked at the quality. So on the X are different types of attributes. And just for reference, most of the metadata I was looking at is the value set one. So all the way on the right. So basically an attribute and a value. It's one of the most popular of the attribute types. And from this figure and from the research that the authors did, it's also one of the most well-formed of all the attribute types. However, you can see that some of the attribute types, even within the value set, are invalid. And basically they deemed invalid values as those that didn't really make sense for the attribute type. So to give an example, if the Boolean, for the Boolean one, if the value is Y, like the letter, rather than like true or false, then it would be deemed invalid. So I think this figure also goes to show kind of how variable the metadata on NCBI is in terms of attribute names, but also in terms of quality of values, which I think is very interesting and helps kind of put my results into perspective as I kind of get there. Now, to get into a bit more depth about the attributes that I was looking at, like weight, for example, here's a table from the same paper as the past few slides. So this table includes concepts, so basically like the group of attributes, so like weight or country, et cetera. And then it includes the number of specific attributes per concept and some examples of those like attribute names. And again, you can see how variable the metadata in NCBI is if there are 26 different attribute names for weight, for example. And this makes the mining a bit more difficult when you can't always predict the attribute name, especially when they're like units are in parentheses or use an underscore, or who knows, maybe there's a misspelled version of weight, or so it's quite interesting. So I'll move on to the actual metadata mining. So I used the RENTRES R package, which is an interface to NCBI's API. And I was able to query NCBI's biosample database for the attributes that I mentioned a few slides ago, like sex, weight, age, et cetera. And then I was able to process the results through R, which was nice because I could clean all of the data in R and then export it as a CSV file. So here is a screenshot of the type of data that I was gathering and what RENTRES was kind of like extracting, basically. I sort of already described some of the information in the biosample database, but I'll just briefly go over it again here. So you can see that this is an entry for sheep and includes sex, breed, and weight. And it also includes that group information, but that is just specific to the study. So it doesn't really provide us with a lot of information. And it's something that came up a lot, where a lot of the values are mostly specific to the study. But in this case, I would query the biosample database for all of the sheep entries, for example, and then could extract out all of the attributes to kind of get the information that the sheep is female, is a Swiss Alpine, and its weight, and et cetera, like things like that. I've also been able to gather the geodatabase information. So you can see it says links, and then it's a link to another database that has more information about the sample and the study. It doesn't include much more information than kind of what's included here, but it would be another kind of reference point. So I have been able to get those IDs out as well through the R Entrez package, which has been helpful. Oh, whoops, I forgot to click the little boxes. There are the boxes. Okay, so now on to results. So there are 4 main results from the metadata mining, and I'll briefly mention them here and then go into a bit more depth over the next few slides. So first, there are about 200,000 returned entries for GBAD species. And again, these were, chicken, sheep, goat, cow, livestock, etc. Second, there were multiple attributes of importance that were present in the database. Third, of all of the weight entries that I showed a few slides ago, there were only a few of them that returned meaningful results. And what I mean by that is that many of them returned metagenomics entries, which, as I mentioned before, we can't use because it's like the clustering of organisms. And then lastly, as reported by that Goncalves paper that I discussed, there's some very inconsistent metadata. So I'll go through kind of all of these one by one. So to discuss the first point, there were many returned results for GBAD species. However, there was some variability. So I'll walk you through this figure. On the X, you can see the attribute name from just GBAD species to GBAD species with sex or age or weight. I also included just weight to kind of give you an understanding of like the general lack of weight information in the biosample database in relation to many other attributes. So, and then the y-axis is the number of returned entries. So from this plot, you can see that there were more entries when sex was included versus age and especially weight. Overall, weight information is not very present, while sex and age information is more plentiful. And I think this makes sense because sometimes sex and age data are easier to track than weight in the field. And one thing that I found for the weight results, many of the organisms were fish. And from my experience, when you're out in the field collecting, especially sex information for fish, if the fish aren't sexually dimorphic or if they're in spawning condition, you usually, have to like physically pick them up to get that information. In that case, it's like just another step to get weight information in comparison to like weighing a cow, So I think that makes sense. Like many weight entries were for fish, so like it's just probably generally easier to get weight information in comparison to some other species. In terms of the specific attributes I was able to find, there were 14 that returned information for GBAD species. And there were definitely some attributes that generally returned more information. So like ID, species, breed, these things were in most of the entries. And that's probably because, those are things that most people, when they're uploading data, just have on hand. But then attributes like sex, age, and weight, condition, and disease were more variable, with only a smaller proportion of entries having actual information. As I kind of have touched on in the last slide. So as I discussed earlier, there's a lot of variability in some of the attribute names. And since I focused on weight information in this project, I've included some of the results from querying NCBI for every weight name that I could think of. I didn't put them all here. I just put some of them here. So on the left side of the table, I wrote down each weight attribute name that I queried. And on the right, I wrote down some of the results. So even by briefly looking at this table, you can see that most of the returned values were for metagenomes, which again, can't really be used because, you're looking at multiple individuals' DNA in one of those samples. However, there were 2 attribute names that returned information for GBAT species, which was weight and birth weight. Weight returned most of the values, but birth weight returned like a smaller proportion of values of entries, but it's better than nothing. And I think this slide is interesting because I think that, even with the attribute names that I mentioned in the Goncalves paper, I also tried to guess some other potential attribute names, but it makes me wonder what information I could have missed if I didn't, guess a specific attribute name that, a certain subset of entries used. But these were some of the ones that I figured might be able to draw some entries. And so that brings me to my last point, which is inconsistent metadata, which I've discussed in depth already. So here on the slide, I have 2 different entries, and you can see that at first they have very different attributes listed. But then you can also notice on the left that some of the values associated with the attributes are missing. And so this was a very common occurrence in the data that was returned with my queries. Not only was like missing there, but like not determined, not included, misspelled version of any of those. Or even I got some weird ones with sex where it was like pooled male and female. I was like, that gives me no information at all. So there was a lot of data cleaning involved in this project because the values associated with the attribute names weren't always kind of related to the attribute names. So even if a query returned an entry matching my query, occasionally that data wasn't usable. Especially there were a few of them that just had like all NAs. I was like, what's the point of even uploading anything if you're not giving us any information at all? So once the data was cleaned, I was able to export it all into a CSV file. So here's just a really small screenshot of one of the CSV files I was able to generate. But you can see that I have all of the attribute names at the top of the columns. And then with the screenshot, you can also see that there's, some variability in the presence of the data where, collection date, location, coordinates, condition and disease are all missing or had invalid values, which is why NAs are all listed there. Either way, since the data are set up in a CSV file, it would be easy to import into a script or into another file for analysis. And these data will also be made available or can be made available with the GBADS API. Okay, so now that I've described the project and the results, I've listed a few limitations here from the project. I've already talked about the inconsistency in the metadata, which is a limitation that would be a bit difficult to fix unless everyone started uploading their data with a bit more consistency, or NCBI changed some of their package upload kind of information. But either way, the variability of the metadata just means that there's a lot of data cleaning involved. Another limitation is the time it takes to run, as that Entrez R package can take a long time retrieving results from a query with many entries. And when this happens, the connection from Entrez to NCBI can fail, returning a failed forwarding request, which can be quite a pain. But overall, I think these limitations are relatively small, which is good. So I'm going to switch gears briefly for my last few minutes, spending a few minutes discussing where I think this work could go. I think it would be interesting to mine other databases for metadata. There are 2 other repositories for genetic and genomic data. The first is the European Molecular Biology Laboratory, and the second is the DNA Databank of Japan. And both are part of the International Nucleotide Sequence Database Collaboration, long name, and all 3 databases, NCBI, EMBL, and DDBJ, are in this collaboration and are supposed to exchange information between the 3 databases. However, this has been shown not to occur as frequently as advertised. So for EBI, which is the European Bioinformatics Institute, which is part of the EMBL, it's basically, from what I understand, like the European version of NCBI, and includes a biosamples database. So that's very similar to the biosample database in NCBI. However, there aren't as many like established packages that can connect to the biosamples database like there is for the biosample database in NCBI. However, we could still connect to like its API and extract information out that way. It's also been shown with that same paper that I was discussing before that EBI has generally higher quality metadata than NCBI. And so I'm going to bring this table back that I had a few slides ago. It shows like the different attribute names, but the table actually included the NCBI biosample records and the EBI biosamples records. And you can see that the number of entries is not equal. So the two databases aren't exchanging information as often as they say they do. And that in some cases, the EBI database holds more information than NCBI. Therefore, EBI might be an interesting path kind of for future work to kind of get some entries that are on EBI, but not NCBI and vice versa. And then the databank of Japan is the only nucleotide sequence databank in Asia. And it's supposed to exchange data with NCBI and EBI, but most likely includes data that aren't on NCBI. So like EBI, we could query the databases and kind of get an understanding of what data are included in this database that are included in NCBI or EBI. So I'll just leave you with this last plot, which shows the amount of sequences on NCBI increasing over time. So on the X is time, and on the Y are sequences, and then basically the number of NCBI web users. So you can see that over time, the amount of sequences are increasing. And I think as sequencing becomes more affordable, we'll get so many more sequences on NCBI. So I think that the availability of these data, especially those that help GBADS, will also increase over time, especially as sequencing costs decrease. So I think it's exciting, and genomics is awesome. And that's it. So thank you for listening. We can discuss and, if you have questions, but yeah.
I have a question, if that's okay.
Yeah.
Nice talk. It was fun to see. I had to upload some data to NCBI recently, and I'm sure it'll be easier for you, but it was kind of a headache for me, so I can understand why there are so many issues with like the metadata. But is there a way to look at? Yeah. Is there a way to look at if for instance, the pigs are laboratory breeds versus production breeds, if that makes sense? I mean, because pigs are a model organism for a lot of experimental studies. So I don't know if those are necessarily the same breeds that are used for production, or if that even will matter for our work.
Right. From my experience with the data that I was looking at, it's so different. Some will specify that and some won't. So it's really, generally, from what I've seen, they don't specify it at all. It would be like a rare entry that, did their homework and really put all the information on there. But there is a way, I think, to at least see if they're more so used in like experimental studies, but in terms of the breeds, it's kind of only what they give us.
Yeah, so that might be more of like when you have your set list of samples or whatever, hand looking through and just from context determining, is this some immune deficient pig model, or is this something that's like real world? So okay.
Yeah.
Thanks.
I had a question.
Yeah.
Sorry, Marie. You can go, Marie. Go ahead.
Sure. Thank you so much for running with this. I'm really glad you've done it, and I'm really excited by it. So you said that most of the species, you've moved off my screen, there you are. Most of the species, there was breed information with the species information. That's really exciting, because I think this is really important for the livestock classification and could make the livestock classification that has to be done for GBADS much, much better. The spatial coordinates that you had, how was that, I've forgotten on your summary, was that commonly there as well? Because that'll really help as well.
I would
Or was
say it was
or
in
did
like,
you have, like, region or country or anything else that we could use as a sort of polygon rather than a specific point?
yeah, so I would say most entries had location, whether that was just a country or a city or like a specific region was totally dependent on the entry, but most of them had some sort of country information. And then coordinates, I would say were in like 40% of the entries, but they were very specific. Like I could open it up on a map and know exactly where everything came from, which was really nice. And in that case, usually those entries also had very specific location information. Like I had the coordinates and then it had like the country, the region, the city or township or, but yeah, it was variable as to how many of the entries had those data.
Yeah. It's great that you've looked up all of those other things though, just rather than what I'd sort of originally mentioned. And you said the nucleotide database wasn't any good then, because that's what we had used previously.
No, I found that most of those data were like more genomic or like the study specific. Like I was getting a lot of like sequencing platform information coming back from that, and not as much like organismal metadata, which is why I kind of moved over to biosample because it was much more specific to the organism.
Yeah, I guess when we'd used it previously, we'd been using it to look at host pathogen, host vector, vector pathogen relationships. And so we were more likely to actually want that sort of level of metadata, but not wanting it to return the address of where the samples were processed. But yeah,
that makes sense.
Different. But yeah, I'm quite surprised that it wasn't a little bit better. The only other tiny thing, and I'm dominating, apologies. Have you found the metadata's improved with time then? Is it much better recently?
Yes, 100%. Yeah, definitely, which I think is like a little hope. Like it makes me happy. I'm like, well, good. Well, hopefully over the next few years, it'll be a lot more detailed. But yeah, I think over the past 2 years, I've noticed, first of all, most of the data came from the past 5 years. And then I would say a lot of the good entries, like the ones that didn't have like missing for every attribute and actually had some descriptive information were from like the past 2 years.
Yeah. And it might be that I think from the conversations that we had about the nucleotide database, they brought in drop-down menus more often in the last probably 5 years or so. So it might be that actually you don't have those problems with synonyms and different names and things.
Right.
If you're looking at that more recent period. Really good though. Thank you.
Yeah. Deb, did you?
I was going to ask, it's sort of related to what Marie talked about with location. I was just wondering, you were focusing on the metadata. I was just wondering, would it be possible to get an idea of where the majority of the samples are coming from? Because you mentioned that like there was the Japan database was the only one in Asia, then you have the European one, then you have the sort of NCBI, which is America/world kind of.
Yeah.
But what I was wondering is, in all of them, do you have are they specializing in regions, or or are there underrepresented regions there? Because is there as an example, is there anything from Africa?
I would have to look into it, but like off the top of my head, there were a lot of entries for like China and that area, and then a lot in Australia, and then a lot in like US, Canada, and even some in South America. But I think maybe Africa was like the least represented based off my memory, but that's something that like we can load the CSV into R and then like do a little bit more analysis that way. But yeah.
Yeah, that I think would be really interesting if we can see what the distribution is, because particularly if we're looking I agree with Marie, the breed information is interesting. And it would be interesting to see though, if we have breed information from a wide range of countries, or are we getting, maybe mostly industrialized farming systems?
Right. Yeah, that makes sense.
No, that's great. I think that this would be really good now to do secondary mining on it, so we can answer a lot of these questions. Really, this is great stuff.
Yeah, and I guess the other thing going off the location is that every entry also had like an organization associated with it, and so every entry did have that. So at least that gives some sort of, because usually it's a university or an organization where you know that location of at least the organization that's funding the work. I mean, that doesn't always match up with the location of where they got the sample, but it provides a bit more information. I think in that case, a lot of it was from like China, a lot of the universities.
Yeah, no, it would actually be interesting to see if there's non-university organizations like ILRI or any like of the livestock organizations that have been collecting data. I don't know if they do that. Like, it would just be interesting to know if they do, because that would be one, even if they're just collecting the samples, that would be great, because, like, it may go somewhere else to be processed. But if they're putting in the samples on livestock, we might then get a broader worldview.
Yeah, that makes sense. I don't know who is next.
Sorry, I just have like a comment. Well, first,
Mhm.
great presentation. That was awesome. And Deb, when you were talking about how many samples were from each country, maybe a density map might be a good idea.
Yeah.
Yeah, my cat's meowing, so I'm
Yeah.
gonna.
They agree. They're agreeing. Something like a heat map, density map, whatever. I think that would be great, just because, just to point out, like, this morning when we were talking to Jonathan in bed, we were talking about data gaps, and they're very worried about that. And, if we then join up all of this information with genetics and genomic information, we may have big holes. Especially when we're looking at some lower income countries, or, in fact, entire continents. It would be interesting, although I would suspect in Africa, I would think South Africa would have some, because they have a really good lab system there. So it'd be just interesting to see. I like the idea, Cassie, of a density map or heat map. That would be great. Another dashboard. Yay! Right. Yeah. They just write themselves. Grace, get one designed. Marie, did you have?
Yeah, I just put a link in the chat. I wonder, there's the Sanger Institute in the UK. So not only do you have universities, but you've got like in the UK, Research Council funded genomics institutes who may have policies and might have, I don't know, their own portals. I just wonder if we've got gaps, if it's worth focusing on some of those. I mean, you might, in the sequences you've got, you might be able to target some of those by looking at the affiliations of where the sequences have come from or something. But I wonder, I mean, there's a guy called Seamus Fanning, who I know who's at UCD in Dublin, who I can put you in touch with. And I know that Shay's, they've got huge numbers of sequencing thingies, and, he might have data which might be accessible. And, there might be sort of certain key people who we can get hold of who might have some useful stuff here.
Right. I've actually seen this institute pop up in some of the organizations from some of the results that I was seeing. So that's good. Not as many as some of the universities, but it's definitely still present in NCBI.
I mean, it's run by one of the big sort of charitable trusts in the UK, who are one of the big funders, and just sort of targeting some of those who are specifically looking at this and probably looking at improving metadata, and who have, projects on AMR, for example, at the moment, and things like that. You might find we get some useful information from those.
Yeah.
And actually, the GLASS database, which is the AMR database. I wonder if something like that, hold on. WHO GLASS database there we go. I don't know whether this is just in people because it's WHO, but there might be. Oh, there's also there's Kern Mintons, who's doing AMR for the FAO. Who I was talking to a couple of weeks ago at a conference, actually, and picking his mind specifically on this. I wonder if they may have some data resources. I think there might be some non-open access or sort of semi-open access ones where we could get some agreements if we can sort of target where you think they're likely to be.
Yeah, and that's an interesting point too, because I think even like with some of my master's work and some of the work I'm doing now, there are a lot of data that's like hidden behind closed doors.
Yeah.
That's hard to get a hold of. Like we're having that issue with one of the other projects I'm working for across the university, where we need that data, but we can't get it because it's like hidden in Parks Canada's like server or something. So that's another thing. Like there's probably a ton of data that we're missing out on just because it's
Yeah.
not on NCBI and it's not open.
Yeah, if we sort of talk about this more, maybe at one of the monthly meetings, Kern, for example, we want to get in touch with the FAO and start working with them a bit more closely anyway. So there's a sort of way in there with him, for example. But you know, there might be some sort of targets that we could make to move this along and try and get hold of some of these more offline resources.
Right.
I think it would be interesting if, go ahead, Teresa.
I've been very patient. Anyhow, really nice job, Cassandra. Thanks for doing that. Obviously, we aren't the only people that have problems with data quality and missing data. First, a silly question. Maybe it's just lack of imagination on my part, but what would a bovine metagenome be?
So I think in the case of the example that I showed you, basically it's either, well, in some cases it could be from like a soil sample or a water sample. I think in this case they just took, like there was probably like a pile of feces and they just took a sample from that.
Okay, that makes
and
sense.
Then you extract the DNA and then you send it off to the sequencing facility and then you could have, bacteria. Yeah, anyway.
Okay,
Yeah.
So now that you've done this, two things. What do you think the true potential is for this to be of use for GBADS? Where would you go next? Or is there something specific you focus on for GBADS? And my second question, and is more to the group, I wonder if what you've done isn't also possibly a basis for a paper.
So I'll answer the first question. In terms of like kind of next, how important I think this is for GBADS right now, I think there's still a bit of work to do, but I think over the next few years, as there's more sequencing data available, I think it'll become more important. Like genomics and sequencing is like everywhere right now, because it is getting a lot cheaper and it's only going to continue getting cheaper. And like now there are labs on campus that have like their own little mini like pieces of equipment, which is like crazy. Like the genomics facility on campus can do like smaller sequences too, which is like 5 years ago wouldn't have been like a big thing. So now I think over the next few years, as there's more data on NCBI, I think it'll become more important or more helpful. Right now, there's so much variability in the data, which I think we could still use, but there's going to be, gaps in the data, but like we've talked about, that's everywhere. But I also think it'd be interesting to look at EBI and the data bank of Japan to see what else they have. Because if it's mostly the same, then focus on NCBI. If it's not, then more data.
Mm-hmm. Do you think there's enough there for us to actually do something with?
Right now, maybe not. Depending on kind of what you want to do with it. I mean, weight information, I don't think so. There's like a lot less weight information than there was with like anything else, but maybe like we were talking about earlier, more of like the location specific stuff might be of use. But yeah, weight was difficult. Like I said, it was like a lot of fish and like rats and mice.
Well,
like,
we might be doing some work in fish too, so.
Oh, okay. Well, there's a lot of fish information.
And well, I just wondered whether this might be sufficient for a paper, or at least an opinion in terms of other uses of genomic data and what are some of the issues, which we're all well aware of.
Yeah, I just put in the chat, we have published on something like this, which Cassandra's already seen the paper, I think. It's the most tedious paper in the world to read. But the usefulness of it was really, really good. And it's very descriptive, but it's in scientific data. So yeah, there's definitely an avenue in which you could publish this kind of thing if you can get enough of it together. Particularly given the impact of this, for example, in the livestock classification.
Yeah, something worth thinking about, Cassandra, because you've done all the work.
Yeah, I think this would be great if we could identify the data gaps around breed. Because in a lot of the data that we have in various places, they're talking about indigenous breeds. And, you know, it would be nice to see if any of that has been sequenced. And, you know, it's in a way, it's a way to generate projects for maybe other groups. Like it would be nice if ILRI, when they're going around doing things in various countries, took samples and sent them off to be sequenced, so that we would have the, you know, like there would be more representation of a, you know, broader number of individuals from a species. Because one of the problems with a lot of bio databases, particularly with genomics, like the Barcode of Life, they have very few samples per type of organism. You know, like they'll have like one sample from a butterfly, one butterfly, and then, you know, like that's it. You know, like, so hopefully all butterflies look like that individual, you know, for representative, because they have actually very few. Like it's shocking how few representatives, like how few individuals have actually been sampled.
Bold is a mess too.
Yeah,
Bold has a lot of data gaps for sure.
yeah.
Even like with some of the reference libraries, it's not to
It's shocking like how many gaps there are. And I would think that if we're looking at livestock, we might find information for the breeds that are popular with industrialized farming. But I bet you there is going to be less for, you know, the breeds where they're just, you know, walking around grazing and, you know, like they're indigenous to that area. So it would be really interesting to actually, you know, try to poke various organizations that are out there in the field to take samples so that they can be sequenced.
I'm pretty sure ILRI's done a lot of genomic work on like trypanosomiasis.
So it'd be interesting to see if they're in the say, like if we could find ILRI as an organization that's in there. Now they might give it to somebody else to sequence, so who knows where it's coming, what the organization would be.
I can search for that though.
It's just that it would be interesting to see if there's that, and maybe if we did like this sort of, as Cassie was suggesting, like a heat map or density map or whatever of the countries that are involved, that might give us a clue about what organizations to look for. Because we know where, you know, like we can easily find out where ILRI has been doing studies and that, or have field, you know, people in the field. But that would be, I think that might be something interesting to propose to Gates, you know, that when they do fund stuff like this, maybe one part of it should be go get genomics, go get samples and put them in there so people actually know about these indigenous breeds.
Or if people are already doing samples, at least submit them.
Yeah, well, it would be interesting to know, because one of the things that I've seen in a lot of papers when they're suggesting what various countries, low-income countries could do to improve their agricultural systems is basically they suggest them moving over to intense industrialized farming. And, you know, so in that, a lot of that is adopt the breeds that are used in those types of systems, right? The chickens are, you know, thing it's always like, go get that specific type of chicken and put it in Africa. And it's like, you know, are we going to lose the information that's there about the indigenous breeds that people have had there for whatever, especially with regards to disease? You know, so
well, there were a lot of projects historically, like 40, 50 years ago, where they put like Canadian dairy cattle in tropical places and they all died. So
yeah. But the thing is, that still keeps getting mentioned every time you see a paper. They, you know, like they still like to mention about, you know, adopting Western methods, and it's like. There's an entire group in Africa that's organized against that, that has newsletters all the time about how they try to fight it. And so one of the things that there's a gap in is that if you're going to compare systems, you need to have the base data. And one of the base things that we need is the genetic makeup of these indigenous breeds. And if it's not there, where is it? Like maybe it is somewhere else. I don't know.
There may be organisations, I've just put a link in the chat again, there may be organisations which are national specific or whatever. There's one in the UK called the Rare Breed Survival Trust, which was set up whenever it was, and they have a gene bank.
Mm.
And I wonder if other countries might have something similar. They're a charity, but I wonder if there are some resources there on a country level again, not terribly usefully, but maybe it is useful. And they would probably know weights and actually have quite good knowledge in the metadata around these things as well.
That's really neat. No, and even something about rare breeds and everything like that. That's neat.
So any further questions, or should we do a group thank you to Cassandra?
Great.
Thank you all for listening.
So other business we have today?
Just a couple of things. Wanted to do introduction to Marie, to William. Actually, Marie, you might be interested in this because we were talking about doing the expert elicitation tool. Meet William. You can wave, William. William is going to be doing this for you. William is our newest member, and he's going to be looking at that for you and Gemma. But I wanted to introduce William Fitzjohn. He's up. I don't know. I don't know where on your screen he is. He's my upper right person. But William's just joined us starting this week. William, introduce yourself.
Yeah, tell us about yourself.
Yeah.
Hi, I'm William Fitzjohn. I'm a third-year computer science student at the University of Guelph. I immigrated to Canada when I was 10 years old, but somehow I still have the accent.
From
the East Coast, place called Colchester.
Thanks.
All right. So
doesn't sound like a very strong accent, William.
It does to us.
It's definitely English, but I wouldn't have said Essex.
Yeah, for sure. Yeah,
Yeah,
definitely. Gone away a bit.
But yeah, we're William's gonna help us out on the dashboards and Docusaurus, but I thought why not put him on the elicitation one to get that software up and going. Yeah, that's his first project. His welcome to the team. No pressure. I just wanted to, I just was thinking, Marie, we were just talking about this. So yeah, William's going to be doing it. We've got him on that, so that's great. The other thing I wanted to point out to everybody is that Amardeep has posted part 2 of the dashboard tutorial, and it's on under winter 2022 on the Slack channel, that Slack channel on our account. So part 2 is there. Everybody rush out and see that. So thank you, Amardeep. Yeah, and we're slowly building up the Docusaurus stuff and everything like that. And also Grace has posted on winter 2022 some of the mock-ups for various dashboards. So everybody have a look, they're great. I really like looking at this. So, let's keep thinking about that. I think they're really good. We're going to start working on some of them. And
so Deb, this morning we said there was something we were going to discuss this afternoon, and I've been in nonstop meetings since then. Do you remember what that was?
is that about something in May?
Oh, yes. Whether someone from our team wants to go to Ethiopia in May, so in a few weeks.
Yeah, I don't know. Anybody putting their hand up?
I'd love to go, but I can't.
Yeah. I think it's a bit short notice.
Yeah.
Because I think it was May 9th, was it?
Yeah.
Yeah.
How about you, Neela? You're closest.
I can think about it and see how many days and see, organizationally, how can I fit that?
Yeah,
Yeah,
let's find out more information about that, because it wasn't very clear to me, and what they wanted us to do there, like, like what the role would be, we'll send Jonathan something.
Yeah, I think it's they're doing the expert elicitation in Ethiopia and wanting to, I'm not sure where the conversations have gone because I've been on leave this week, wanting to recode the Cook expert elicitation process into R or something, possibly. So I think maybe Philip Rasmussen and Gemma, and I don't know who else because I've not been here this week, I'm sorry. I think maybe they were going to try and be there for a week or 4 or 5 days or something in Addis working together.
Well, actually, that's what we're hoping William will have done before they go.
Oh, okay.
So we're not going to recode. Yeah, we're not going to recode it, but I knew you were interested in that, and we met with Helen and Gemma.
Yeah.
When was that? I don't know. Maybe yesterday.
Yeah.
Matthew.
Hey, Matthew.
I think. Yes. Sorry, we're just wrapping up. I think you must have had the timing off by an hour. Did you change times in the last week or something? Yeah. Oh gosh.
But you know what? It's okay because we recorded it.
Yeah. Okay. I'll get you the recording.
Yeah, and I'll post. We'll get the recording out. But we know you're interested, so this is good. So we'll send you the link as soon as I get the video processed.
It's a shame. It was a good presentation.
Thank you. Thank you.
Yeah.
Okay.
You'll see it. We'll get it out to you. But and we'll include all the questions in the discussion because I think the discussion was really good after it too. So we'll get all of that there. So, but thank you for joining. That was
nice to see you.
Yeah.
So for the rest of us, Matthew, can you tell everybody who you are? Because most people here haven't met you.
Oh, hi, everyone. My name's Matthew Stone. I'm a veterinary epidemiologist up until recently working full-time at the World Organization for Animal Health, now consulting to them, based in New Zealand. And in this project, I represent the World Organization for Animal Health on the Operational Management Committee and the Steering Committee.
And Matthew's been a great advocate for us at the OIE. He gets this, so helpful to have somebody on the inside that appreciates what we're doing.
Yeah, so it's too bad because we had a good discussion with that, but I'll make sure that I'll have the whole video so that you can watch that. And if there's anybody else, I'll send you the link and you can distribute it to anybody that wants to see it.
Yeah, sure.
Matthew?
Definitely my bad because we did just change out of summertime recently, and I didn't shift the appointment.
Well, yeah, we'd hope that the technology would do these things for us, but sometimes it messes up. Yeah, Matthew, are you going to ISBE?
Yeah.
Okay, great. So one of the last things I wanted to mention to everybody, we're taking 9 people to ISBE, and initially we had I made everybody book early so it cost less. And we were supposed to have a GBADS meeting before ISBE, but it's now been moved to after ISBE. So I just wanted to tell everybody, have a look. If it costs more to change your flight than it would to stay a night in Halifax, it's up to you how you want to go about that. But Jonathan has now confirmed that the GBADS meeting will be the day after ISBE. And I know a couple of you are going on to PEI for some of the courses. So just plan on staying as long as you can on Saturday and driving later that evening. There's going to be a dinner too, but you probably don't want to wait to drive the next morning. It probably wouldn't give you enough time unless you really want to go to bed early and get up early. Anything else on that?
No, I think we have a lot of, we've got 3 oral presentations and how many posters?
I know we had 9 submissions in total, so
6 posters. Okay.
yeah.
We have 6 posters and 3 oral presentations. So we're going to be
In
busy.
addition to the special session on GBADS. So GBADS will be well represented.
Yeah, it's going to be good.
Okay, do we have anything else for today? Anything now that you have us, Matthew? Anything you'd like to ask or say?
No, but I'll catch up on the recording.
Okay, nice
All
to
right.
see you anyhow.
Okay,
Yeah.
see you.
And just to everybody, you can go to the team meeting Google Doc and fill in anything about what you want so we can catch up with that. And next week, we'll just have a regular meeting where we'll just catch up on everybody's what everybody's doing. And if people have demos or presentations, that'll be good because we like demos and presentations. It's always fun. So
Grace and
yeah.
Cassie have questions.
Oh, well, I just wanted to say the one thing I had to bring up today is that a couple of us have worked out like a blog schedule for and like template. So I linked to that in the weekly update in my section. So if you guys want to read it and comment or sign up for a time, that would be great. But it's in there.
Awesome.
Otherwise, we can talk about it next time. That's it for me.
Thanks, Grace.
Oh, great, Grace. That's great. The blog schedule. We've got presentation and publications going up. All right. This is great. Okay. Actually, let's talk more about that next week too. Let's sort of focus on that because I think that'll be great, particularly too, because we're getting ISV stuff. We're thinking about that. And there's this side data con. Did I get it right, Cassie? So we've got that four of us are giving talks or abstracts, whatever. So we've got that. So we've got a lot of stuff coming up. We've got a lot of things that we need to get on our site, publications, presentations, everything like that. So this is great. Why don't we talk about that next week as sort of our number one sort of discussion item? And then we'll go through. Yep, go ahead.
Cassie is next, and then I have one question I'd like to ask. I just got something in from IDEX today. They're asking about, anyways, I'll just read the question. I have to go back and look at it. Okay, go ahead, Cassie.
I just was wondering if I have to submit slides to the One Health Symposium conference by April 29th. So I was wondering if I could, when I make those up, hopefully by the end of next week, if anyone would like to review them for me based on the abstract, because based on the abstract, we're kind of connecting GBADS as well as some of the other global burdens. So it'd be good to have.
Why don't you present that next week and we can go through it with you?
I could do that, yeah. That puts me very ahead of the game, but I think that I can put them together. Or at least something preliminary.
Yeah, it doesn't have to be perfect, but yeah, that would be good. We can do that. We can talk about the documentation site because there's lots of the DocuSource site, there's lots there. And maybe Curtis, we've been talking about like the color scheme, like the sort of the design scheme. Maybe we can discuss a little bit about that, because that will also be something maybe we want to also put on DocuSource to make sure all the colors, fonts, and everything like that look the same as our presentations, which look the same as our dashboards, that sort of thing. Maybe we can discuss that as a group. You can lead that discussion.
So I just put this into, did I send it? Does everybody see that? Or did I have to hit something yet? There we go.
Yeah,
Okay.
there we go.
So rather than, well, I'm just going to read it. But so I just got this from somebody at IDEX. Somebody at IDEX connected me to somebody else who's a veterinarian, but has something to do with data. And they're wanting to present more on their AI-powered disease prediction work and get IDEX's names in conversations about AI and have a couple opportunities to speak about their capabilities related to vet med, but not necessarily to vets. I mean, ISV would have been a good place for them to go, but it's a little too late for that now. So I wanted to ask the computer scientists in the group if they are aware of anything as well, well, probably health informatics like AMIA or, okay, I just thought I'd throw it out there in case I can think of some health informatics places they could go. Are there any computer science things that are focused on AI applications in health that you're aware of? Deb,
There are,
Andrea?
I mean, there are. There's informatics, there's AI approaches to medicine or, biomedical stuff. Andrew, do you know of any specific? I know a few in Europe.
Yeah, I'm thinking about EMBC as well.
If you just want to send those to me, then I'll put it together in a response.
Sure. I think their timing is really bad because I think all of them just actually closed or occurred.
Yeah, well, same with ISB. But I mean, there's next year, I guess. ISB's 3 more years, but anyhow.
There may be
Yeah.
some opportunities at some European conferences because there's a bunch of them that go in around the October,
Right.
November timeframe. So a couple of them may still be open, you know,
Okay,
like for papers. I'll look it up. I know a couple of them, so I'll look up and see if that's a possibility.
maybe something like the Society for Veterinary Epidemiology and Preventive Medicine in Europe, which usually meets around March or April. So if you have a few things, send it to me and I'll craft a response to them. I
Okay.
just thought I'd take this opportunity to survey our members here. Thanks.
Hmm.
Okay, and yeah, for everybody else, you know, I think we have a lot of stuff to discuss next week. I think it'll be good. I think that'll also get people being able to, you know, take what we're talking about next week and contribute to it or use it, that sort of thing. And one of the things that for everybody to do is go through Emerdeep's tutorials, right? So about the dashboards, and you know, we can chat about the design and branding and colors and fonts and all that next week, and put that for the dashboards and for DocuSource.
And Cassie, for the One Health thing, you might want to go back to what we did for what we did with Graham Taylor. What was that? One
Yeah,
Health.
the complex system.
Yeah,
data
that would be a good
collection.
starting point in terms of seeing what we had there. And then, how long is your presentation?
11 minutes.
Yeah.
Yeah.
It's
which
always
is
harder.
kind of a random number, right? 11
Well, it's
minutes,
probably
4 minutes
then-
for questions.
exactly 15 minutes. Yeah, that's very similar to ISB. So, okay. Well, we accomplished a lot today in our various meetings. So I guess we'll see you next week, except for those that we'll see at 4 o'clock.
Yeah, and everybody filling their stuff on the Google Doc, and we'll keep going.
Okay,
All right,
go
sounds
team.
good.
Thanks again, Cassandra.
Yeah, great talk. Great talk.